So I'm pleased to be hosting Bess Hardwick and Deirdre Kerdraon to discuss the recent PLOS ONE Lab Protocol paper entitled LIFEPLAN, a worldwide biodiversity sampling design, and the associated detailed protocols in protocols.io. Bess is a biologist who has worked in ecological research projects at the University of Helsinki, often on distributed science projects. She's currently a project planner at the University of Helsinki, working as one of two coordinators in the LIFEPLAN project. Deirdre is currently project manager at the Swedish University of Agricultural Sciences and one of two coordinators for the LIFEPLAN project. Before this, Deirdre did a PhD in environmental science looking at plant soil dynamics in forests. Deirdre and Bess, thank you so much for talking to me about your protocol.
Thank you. Nice to be here.
Thanks for inviting us.
To kick off, can either of you briefly explain what the protocol or protocols actually are about, how to use them, and why these protocols are relevant?
So the protocol is related to a larger project called LIFEPLAN, which is a global terrestrial biodiversity research program. We collect biodiversity samples around the world, and we identify species in them with molecular methods and machine learning methods. And then we develop new statistical methods for modeling global biodiversity. And this protocol is about the field sampling methods. And the field sampling is carried out by independent researchers all around the world. They've been collecting samples since 2021, and they're still collecting samples and will be collecting until the end of this year, until the end of 2025. And we use 5 different methods. There's malaise trapping for insects. Then we use cyclone samplers to collect fungal spores from the air. We collect soil samples, which we also sequence for fungi. And then we do camera trapping and audio recording. So there's 5 different methods that are described in the protocol. And the reason, the way we use the protocol is that LIFEPLAN it's a method. So it's a standardized method that we would love for other people to take up after LIFEPLAN ends, to collect biodiversity long-term data sets in a standardized way. And publishing it as a open access protocol, it makes it findable and accessible, obviously free to use and standardized. Also, it's just very practical for us when we're writing papers that we don't have to write the same methods over and over again, because we're going to produce lots of publications that we can cite a fully published and set protocol.
And to further understand the significance, I do get the ecological implications of the protocol, but technically speaking, to further understand the significance, can you elaborate on which protocols were already available when you embarked in this project and why those protocols didn't work for you? Like, in other words, what was the motivation to optimize and put together these protocols?
So usually when people publish new protocols, it's because you're using them with the latest technologies and the latest methods for their specialty subject. We actually had to do the opposite. Because all our sampling is all over the world, we actually needed to go back and design protocols with the simplest possible solutions and taking into account a complete lack of infrastructure. So unlike a lot of, unlike most protocols which are there to describe the novel, what's novel about what we do is the fact that we're sampling these 5 different methods, and that we're doing it in such a way that it should be able to be done with minimal electricity or with limited access to water, or in Antarctica, or in the desert or in the jungle. And so for that, there have been many protocols, but we've really gone back to the basics.
I would like us now to go a little bit deeper into the developing of the protocol. And can you tell us what were the highs and lows on developing these protocols? Is there anything, as you were putting these protocols together and, let's say, simplifying the protocols, was there anything that you tried that didn't work and didn't make it to the final version of the whole project?
Well, we developed the protocols together with the teams all around the world. So we can send them draft versions and plans of what we would like to do, and we got lots of feedback about things that wouldn't work. I think there are a lot of things that got dropped along the way. We were initially planning to collect root samples as well for fungi, but picking which plant species to sample from all over the world, that turned out to not be feasible or standardizable. But yeah, learning from the teams about the very different and difficult conditions that people do field work in, that was really interesting. In terms of highs and lows, I suppose most fun was testing cameras. Camera trapping is also always fun. I have lots of funny pictures of professors jumping in front of cameras in the middle of the night in the forest, trying to run really fast past it and things like that. I suppose the most difficult bits were standardizing soil sampling. There's no such thing as a standard soil sample, as Dee will be able to tell you a lot about. But another one was for the audio recording. So figuring out those are limitations of batteries and memory cards. Like, we wanted to record as much as possible, but then we want to make sure that the batteries and memory cards will last in minus 30 degrees Celsius and in all kinds of moisture conditions. We did end up having to reduce the amount of audio recording because we were just generating so much data that we couldn't transfer and store it all. So that was one change that we did have to make. Also, actually, halfway through the project, we had to reduce the number of recorders and cameras on each plot, just because the equipment was breaking down even faster than we thought it would. So everybody did tell us that camera traps don't last long in the tropics, but it was still a surprise to see how much they break down.
This is a big project. You know, the paper contains, in fact, 5 independent protocols to sample from the air and from the soil and sounds. So I don't know how easy it's going to be to answer the following question, but if people want to use the protocol, what are the most critical steps? And what parts are particularly tricky and others might make mistake on? You already mentioned something about the cameras and the storage that you need to maintain the data. Can you give us other parts that might be critical?
Yeah, so actually the way we've written the protocols, because we tested a lot of them, I think themselves, they're really well written, and so you can follow by step. But I think the really important part and the really critical step is how you record the metadata for each sample you collect, so that all the samples collected can be reused in the future, because part of our project is to, yes, we're collecting samples now and we're collecting data now and we're analyzing it, but we're also archiving some of the samples in the hope that in the future, when technology is improved, then we can sequence or analyze more data from those same samples, so that we actually have a picture of the environment between 2021 and 2025. And we're thinking, for example, we're asked a lot about plant DNA, which we weren't able to do, and hopefully in the future someone will be able to access those samples and sequence those.
This is a multi-year project, and you published the protocol paper about 6 months ago or so. And you already mentioned, Bess, something about, you know, having to make changes as it went, because, you know, you didn't realize the amount of, for example, data that you're going to be collecting. Has there been any other optimizations or changes to the protocol since the time of publication?
Since publication, no. So we published the protocol once it had settled and once we decided that, okay, this is now the version that works. And the idea is that this is now the final published one that we are sticking to. It's final version 7. This is the one. So not since publication.
I would like to change this gear now a little bit, and you put together this very well written set of protocols, but then you decided to take them to be peer reviewed and published as the standalone methodological paper in PLOS ONE. What was the motivation to have the protocols peer reviewed?
We think it was the best way to combine the very diverse field methods and also to take the time to actually describe all the field sampling. And so to have it actually approved and read over by other scientists when the aim of this LIFEPLAN methods is to have it used by others is the best way to promote the work and to also confirm that what we're doing is actually worthwhile. Do you have anything to add, Bess?
No, not really. That's it, I think.
And how did the feedback that you received from your colleague peer reviewers shape the final version of the paper and the protocols?
I think the input from the peer review was very much what it always is with peer review, that it clarified and improved the paper quite a lot. When you're writing from inside the project, lots of things seemed obvious to me and Dee, but then of course when the reviewers read it, they ask, can you clarify this and that? And I think that improved it a lot. They did actually also find one mistake in the figures that I'm very glad that we corrected before publication. And yes, they also helped us to clarify the rationale behind the whole protocol and why we're actually doing it. So it's always really helpful to get that outside expert help on that. So the peer review was very helpful and valuable.
Why did you decide to make the peer review history public together with the paper?
Well, very simply is we believe it's a step towards making scientific publishing more transparent.
And my last question would be, the paper is relatively young, so this might be a speculative question at this point, but what do you think has been or will be the impact of this methodological paper for both of your careers, considering the different stages at which you are?
Well, like you said, that remains to be seen. Well, I think it's a nice thing for our CVs, and we'll see. LIFEPLAN is going on until the end of March 2026, and we'll see what the job market is like. But I think we both would like to continue working on these kinds of projects, and it's a nice little demonstration and merit for us to show that we've done something.
I agree.
Well, I don't have any more questions. I don't know if there's anything that you would like to add that haven't been discussed so far.
Just that I think publishing methods, especially with the way journals are these days, that they increasingly want shorter texts, and publishing your methods in a standardized way with something like protocols.io, where it really prompts you to think through everything and put in every single detail, like what is the unit of measurement on every lab chemical and every single thing. That kind of standardization, I think, is very helpful. And having a DOI and having a thing that you can point to and cite is very useful. So I would recommend the process to anyone.
Yeah, I second that. I also like that it makes it a lot more reproducible because you can really have a step of method. So if someone does come and think, okay, I want to reproduce this, then it's not just a small paragraph of methods where you have to guess all of the in-between sort of minor steps. So for that, I think it's very good.
Bess, Deirdre, thank you so much for your feedback. Thank you so much for your insights. It's been really insightful.
Thank you.
Thank you.